Hi, it's a real pleasure to welcome you all this evening to our second session of the Science series. So thank you so much for all of you joining us for this session on cancer vaccines and oncolytic viruses. It's also my pleasure to introduce you to today's speakers. We've got Katie Cambrook and of Counsel in our patent litigation team, who's going to cover the cancer vaccines part of the session. We've got Erik Müürsepp, who is going to cover the oncolytic viruses part. And finally, last but not least, and for those of you who were here 2 weeks ago, Louisa Jacobs is going to cover the legal aspects of these technologies that we're going to talk to you about today. So I'll hand over maybe to Katie first. Thank you.
Hi, so as Jessica said, I am Katie Cambrook and I am here today to talk about cancer vaccines. I think between Erik and I, we're going to speak for about 45 minutes or so before we hand over to Louisa. So first of all, just to set the agenda for my kind of section of the talk, we are going to start by doing a brief history of the kind of cancer vaccine scene, particularly looking at the therapeutic side. Then we're going to look at the types of vaccines that are available, preventative and therapeutic. The majority of this talk is going to focus on therapeutic cancer vaccines. So after we've done just a brief look at preventative vaccines, we'll go on to look in a bit more detail about therapeutic vaccines, their mechanisms of action, advantages, limitations, kind of where we are now and where we might be heading. So to start with on the history, as you can see a little timeline on the slide, the history can be traced back to the early 1900s or the late 1800s, depending on what you read, but it all seems to be consistent that it was a man called William Coley who used inactivated bacteria to try and kill tumour cells, and there was a kind of regression of the tumour that was seen. Going on into kind of the 1950s, there was some work done with BCG, and again, that was used to infect mice cells in this instance, and there was some tumour regression seen. Rolling on to the 1990s, and BCG was approved as a cancer vaccine by the FDA for the treatment of a particular type of bladder cancer, non-muscle invasive bladder cancer. We roll forward into the 2000s, and we see more companies coming into the fore as kind of technology and science moves on, and you see companies such as BioNTech coming in and looking towards kind of a more personalized medicine that started to come in that kind of era. Then in recent years, we've seen quite a surge in interest, and that follows the sort of approval and widespread use of other cancer immunotherapy, things like checkpoint inhibitors that kind of take the brakes off the body's own immune system and allow that to kind of work to treat the cancers, and also following quite a lot of the vaccine work that went on during the pandemic. So, the types of cancer vaccine, as I mentioned before, there are two types: preventative and therapeutic. The preventative vaccines are those kind of vaccines that we would normally kind of take for flu and things like that, and they're given to try and reduce the risk of someone getting a cancer. The therapeutic ones are given to someone who already has cancer, and the aim of those is to boost the body's own immune system to target and then kill the cancer cells. So, on the preventative side, there are a couple of vaccines which are quite well known, shown on the slide. One is in relation to human papillomavirus. That's been around since around 2008 and is now available to all school children aged 12 to 13. And it protects against a number of different cancers, but in particular, the most widely reported one is cervical cancer. And together with this vaccine and a kind of the usual screening program run by the NHS are actually hoping to eradicate cervical cancer by 2040. So these vaccines really do have a great impact on public health as well as everybody's individual health. The other example on the slide is the hepatitis B vaccine. That is a vaccine against hepatitis B, and in particular a chronic infection of hepatitis B, which can lead to particular types of liver cancer. Again, it's now a routine vaccination that's given out to babies. And again, there are aspirations this time from the WHO to use this vaccine to eradicate hepatitis B from the world. So we're going to move on now to talk a little bit about therapeutic cancer vaccines. And before I do, and also to kind of introduce some of the things that Erik will talk about in his talk on oncolytic viruses, we're just going to have a little intro to the human immune system. So it's made up of two sides, the innate and the adaptive. The innate immune system, as you may well know, is the system that we are born with. It's a rapid kind of reaction squad within the body that can really quickly target infections which are recognized inside the body. That usually takes place in the form of viruses and bacteria and other foreign bodies which may come in. It's in particular recognizing particular antigens, and that's what activates this immune system. The innate system is non-specific, so and it doesn't have the ability to form any memory. So while it acts really quickly to hopefully do the job, it can't retain where it's seen a particular antigen or virus before. These antigens are just particular types of proteins and things, and they're often expressed on the surface of different cells, so bacterial cells, and that's what helps the body recognize that they are foreign. On the other side of the slide, we have the adaptive immune system. That's a far more specific system. It's a little bit slower to react, but when it does react, it recognizes a specific antigen. And on that basis, it can be quite targeted in its response. The antigen is presented to the immune system, so the cells in this side of the immune system, for example, the T cells, by an antigen presenting cell. That is a typical antigen presenting cell is a dendritic cell, kind of shown on in the innate system. So there is interaction between these different sides of the immune system. The antigen is kind of recognized by that dendritic cell, as I said, taken up inside it, processed and pushed out onto the dendritic cell's surface or another antigen presenting cell in a particular complex which can be detected by these T cells and the receptors on them. And that activates the T cells to then go about their business of affecting an immune response. So, in most of the cancer vaccines that we're going to be looking at, it's this kind of T cell response which is what is being looked at. There are other responses, obviously from B cells and other aspects of the immune response, but it's the T cell response that we're going to focus on. So this screen slide shows what we've already discussed about activating the adaptive immune system, antigen presentation, activation of T cells that then goes on to cause this cancer cell death, which is the affected immune response here, and also forming an immune memory. To focus on a couple of those points in a bit more detail, the antigens that we're talking about in the terms of cancer vaccines are particular tumor cancer vaccines that are expressed by the tumor. These can be tumor-associated antigens or tumor-specific antigens. And you may also hear the terminology neoantigen, which is a specific antigen that is expressed on the surface of cancer cells, and they can be different between different people. Cancers, as you will probably know, are very heterogeneous. Somebody's pancreatic cancer in one person will not necessarily be exactly the same in another. The cells within the cancer can mutate in a different way, and so they present on their surface these different individualized, almost neoantigens. And so it's these antigens which are being looked at and taken because they are often only expressed on the cancer cells and not in other cells. And that allows the cancer vaccines and the immune system to specifically target these cancer cells. We'll go on to look a little bit more about how the T cells cause cancer cell death, but just to mention as well about the immune memory formation, which is on this slide. As I mentioned, the innate immune system cannot form a memory, but the adaptive system can. And there are particular types of T cells, memory T cells, which are retained within the body, and they can be reactivated and circulate in the body, which can try and help prevent the recurrence of a cancer. So, when it comes to looking at these different antigens that I mentioned, which are expressed on the surface of the cancer cell and which are packaged up to be made into the vaccines, which can then be recognized by these T cells, we have a couple of different options that have been available and which we are moving towards more in this form of the personalized vaccines. So taking first of all the off-the-shelf vaccines, these slightly crudely named, but are kind of what they sound like. The idea behind them was that you could select particular antigens that could be specifically expressed and then targeted from, say, a prostate cancer or a melanoma, and they would be common among a group of different patients who all had melanoma or prostate cancer. You could put them in a vaccine, it would sit on the shelf, someone would come in and be diagnosed with a cancer, and that kind of vaccine could be used. The problem with that is, as I said, that lots of the cancers are very different, and there was quite a lot of off-target reactions when this was used, and not great efficacy. So now, a lot of the vaccine work that's happening is moving towards the personalised side, and in this production of vaccines, a sample of a person's tumour is taken from them, analysed. They look at the neoantigens that are expressed and presented by that tumour, and they look to see which ones may elicit the strongest immune response. They take them out, they package them up into a vaccine, and they put them back into the person. That's kind of more the direction that things are looking in at the moment. Whether or not, through looking at all of that personalised information, we get a better library that allows some more off-the-shelf vaccines to be developed, we'll wait to see. So there are a couple of different types of vaccines which I was going to touch upon today. The first of these is the peptide or protein-based vaccines. These, in some ways, are the simplest type of cancer vaccines that are available. They are essentially take the protein or the peptide, it could be the antigen, out of the cancer cell, or it can be made artificially within a laboratory following kind of, looking at libraries of well-known cancer proteins. It's then injected back into the patient. When that happens, it is recognized by these antigen-presenting cells and other immune cells that circulate around the body. It's taken up by the antigen-presenting cell, and as we discussed before, that antigen-presenting cell processes that antigen, pushes it up to the surface. It goes off to the lymph nodes where the T cells are. The T cells recognize that specific antigen and get activated. Dendritic cell vaccines are a little bit more complicated, but follow a similar kind of pathway. They typically see dendritic cells from a patient being removed or done at least outside of the body. Dendritic cells are exposed to a number of antigens. They are then taken up by the dendritic cell. That dendritic cell is packaged into a vaccine and then put back into the patient. That dendritic cell, obviously already being an antigen-presenting cell, already has on its surface the antigen presented ready for the T cell to come along and recognize and then be activated. There are some moves at the moment, because that's a particularly expensive way of making vaccines, to activate dendritic cells within patients' bodies and couple dendritic cell vaccines with kind of traditional methods where cancer cells are killed and the antigens which are inside them or on them are released inside the body, and the dendritic cell which has been activated then can find that antigen, recognize it, take it up, and then go on and do its job along with the rest of the way, how the others have worked. I just want to spend a moment or two as well on DNA and mRNA vaccines, which particularly from the mRNA vaccines, we've all become quite accustomed to hearing about some of those from the COVID days. In these types of vaccine, there's kind of two different pathways. So starting with the DNA vaccines, they package into the DNA the genetic material that reads for particular antigens or proteins. They're put into a vaccine which is injected into the patient. The package has to then make its way into a cell. And inside that cell, it then has to go into the nucleus of a cell where where transcription and then takes place. The DNA then goes outside, well, the messenger RNA then goes out of the nucleus into the cytoplasm, and a protein is made. So there's a little bit of additional steps to get to the protein section where we were before. The protein then inside the cell has numerous different ways of kind of triggering the immune response. It may be kind of pushed outside of the cell through different cellular processes, or it may be presented on the cell surface itself, so that cell becomes an antigen-presenting cell. For the mRNA vaccines, they tend to be now trying to be packaged, at least, in a way that they are selectively taken up by dendritic cells and other antigen-presenting cells, so that they go straight into there, and then the process of making the protein is happening within inside the dendritic cell, and they're then pushed out onto the surface using the usual complexes to be identified by the T cells and so the T cells can then go on and do their business. So we've talked a lot about these activated T cells. What do they actually do? How do they kill the cancer cells? So once they are activated, they then migrate. As I said, they activate in the lymph nodes. They migrate to the sites of the tumor or the cancer. They identify the relevant antigen that has activated them previously through the vaccine, and they release a kind of one material which opens up some pores within the T cell, and another material which is a kind of cytotoxic package which goes into the cancer cell and causes that cancer cell to die. So we talked already about the BCG vaccine, which is around and has been for a number of years now for bladder cancer. Another example of a vaccine which is around is Provenge. It was approved in 2010 for particular types of prostate cancer. It works by taking the patient's own cells out, exposing them to an antigen and then putting them back into the body so that when they are back inside the body, they can recognize the antigen from the cancer and therefore act upon it. It's a particular protein antigen that they are exposed to, PAP, prostatic acid phosphatase, which is a particular protein or antigen which is activated in cancer cells. So it's a more generalized approach than the personalized approach. But what you can also see on the side there is the cost of the treatment, 93,000 for a course. So what are the advantages of therapeutic cancer vaccines? So as I mentioned, they are targeted and specific. So unlike chemotherapy and radiotherapies, which affect healthy cells and can cause damage outside of the tumor region, therapeutic cancer vaccines should be targeting just the cancer cells and causing them to die. The approach, as we've mentioned, is also or can also be personalized and looking at particular neoantigens that are presented on an individual's tumor and then creating a vaccine which is personalized to that person. We hope that there would be a durable immune response, as we've discussed, with these memory T cells, which will be circulating afterwards and hopefully helping to prevent recurrence. One of the advantages as well, and possibly a direction we will look at things could be moving in, that's how cancer vaccines can be used with other therapies. So there's kind of two angles to this. One is that they could be used to treat potential residual disease or as a kind of adjuvant therapy. If someone's had a tumor mass removed by surgery, a cancer vaccine could be used after that to try and target anything residual that's left behind that you can't take away because it's too intricately linked with blood vessels or so on. And it could also be used in synergy with other therapies, and in particular the kind of checkpoint immune checkpoint inhibitors, which we mentioned before, the two of them working side by side. So from what I've said, it's complicated, but once you've got the T cell activated, it sounds like it might just migrate to the tumor site and do its work, and the cells will die, and where's the question? But there are some challenges and limitations with therapeutic cancer vaccines. We've mentioned already the tumor heterogeneity and antigen selection. How can you identify the correct antigen? Can you make vaccines that can work, that aren't just personalized, that can be used for different people? Who do you select when it comes to patient selection? And what is the timing of a treatment? Cancers respond well if you can treat them earlier on, but how can you make sure you've got the right antigen? What's to say that it won't mutate further and that one treatment that was working then won't work anymore? How can we think about how that should be dealt with? There's also a need to think about combination approaches, which I talked about before. That's going to need a little bit of careful thinking as to how those different approaches fit together and how different compounds that act on the cancer cells or on different parts of the immune system might work together. There's also the fact that cancers are very sneaky. They haven't got to be cancers without evading our own immune system in the first place. So when these activated T cells reach the tumor site, they can find themselves in an environment which is already immune suppressed. And therefore they might not be able to carry out their action quite as effectively or at all to kill the cancer cells. They might also find that the antigen that was once presented, as we said, either mutates or the cancer cell sucks it back inside, doesn't present it anymore. They find their ways to evade cell death, and that's something which will need to be also looked at. Last but by no means least is manufacturing and cost, how these vaccines can be made on potentially smaller scales, but at no less cost if you're doing personalized vaccines, and the controls and regulations that need to be put around that. Again, we've got some learnings from what's happened already in relation to mRNA vaccines, but there's still a lot to be looked at. But to end with some future directions and perspectives and a little bit of hope, there's a couple of things which I just put on the slide here as things to watch. And the first of those is a combination of a neoantigen vaccine, Moderna's mRNA 4157, and Merck's Keytruda, which is one of the immune checkpoint inhibitors that I've mentioned, which is currently in phase 3 clinical trials for the treatment of melanoma. This is a worldwide trial and follows positive results which were announced from phase 2B trials at the American Society of Clinical Oncology earlier this year, where the combination of the two showed a reduction of 49% in death or recurrence over the treatment with Keytruda alone. So hopefully results expected by 2029. The other one to note happening here, particularly in the UK, is that last year BioNTech and the UK government entered into a partnership or agreement, which has resulted in the NHS's cancer vaccine launchpad. That's testing personalised mRNA cancer vaccines with the aim of testing it in up to 10,000 people, or treating 10,000 people by 2030 with these vaccines. So again, that's going to give us a wealth of knowledge, which will hopefully help towards progressing these as we move forward. One thing I haven't touched upon in great detail, although I mentioned it in relation to the mRNA vaccines a little bit, is how these vaccines are packaged and reach where they need to reach within the body. This kind of advanced deliveries technology is going to be something which is going to be at the fore of getting the vaccines packaged up in a way that can reach the target and can also be made in an affordable manner. And I think that's one of the avenues which Louisa will touch upon towards the end as well. So with that, I will hand over to Erik.
Thanks, Katie. So I'm going to spend a bit of time talking about a type of cancer therapy which you may or may not have heard about before, and that's called oncolytic viruses. So we're going to have a brief look at how this treatment option has developed over the years. We're going to have a look at what a virus is and what distinguishes an oncolytic virus from a bog standard virus, how it goes about killing cancer cells, what some of the challenges are, where we are at the moment, and also where we might be heading in the future. So let's have a look first at how oncolytic viruses developed. And the story is somewhat similar to cancer vaccines that Katie mentioned. So it started off in the early 20th century with an observation that doctors noticed something relatively unusual, that some cancer patients who caught viral infections or who were vaccinated using a virus, they sometimes had their cancers shrink. And this prompted quite a bit of interest that led to some animal studies in the 1920s and some early human studies in the 1950s, where they took some cancer patients and they injected live viruses to them. But those early human studies were quite inconclusive, and as you can imagine, they were often quite unsafe as well. The real turning point came in the 1990s, and that was with the advancements in genetic engineering, because from that point onwards, researchers had tools to modify viruses to selectively target cancer cells, but without harming healthy tissues. And we could therefore create sort of more safe and effective therapies, which then led in 2015 to the FDA approving TVEC as the first approved oncolytic virus on the market, and quite a bit of interest and research in this area since then. So let's consider what we mean when we talk about a virus. Viruses are tiny infectious agents. They're much smaller than even bacteria are. And they need a host cell to survive and to replicate. Unlike cells, viruses aren't really alive on their own. They can't reproduce, they can't carry out metabolic functions without a host. And the structure of the virus is very simple. It's just a bit of genetic material, either DNA or it can even be just some RNA, which is packaged up in a protein shell, which is also known as a capsid. And some viruses also have this sort of lipid envelope surrounding the capsid, and that's actually a remnant of the host cell membrane, which the virus sort of grabs along with it as it's bursting out from a host cell. And the way viruses work is kind of like a hijacking operation. So they attach to a specific host cell using those proteins on its surface, and they recognize and bind to specific receptors on the host cell surface. The virus then gets taken up by the host cell, and it injects its genetic material into the host cell. And that genetic material takes over the host cell's cellular machinery. And it basically starts churning out more viral particles. It turns into sort of a virus manufacturing factory. Those viral particles assemble, they burst out of the host cell, and they go on to infect other cells. And that's how the virus sort of can spread quite effectively throughout the entire body. And often although not always, the host cell dies in the process of this taking place. So why do some viruses have this sort of natural ability to kill cancer cells? Well, our cells are actually really efficient at fighting off viruses. It's why most viral infections don't really last for very long, and we're usually able to overcome them. But cancer cells are different. So cancer cells have accumulated a number of mutations, and the primary effect of those mutations is to just make the viruses the cancer cells quite similar to viruses. They just want to sort of replicate and make more of themselves. That's the sort of the purpose behind the mutations and the purpose behind the cancer cell. But these various mutations, these sort of weird things happening in the cellular machinery, also mean that cancer cells are very bad at fighting off viral infections. So a virus which any of our normal cells would be able to kill quite effectively is much more effective against a cancer cell, because a cancer cell, the priority is just to self-replicate. Its priority isn't to fight off viral infections. And we can exploit that sort of inherent vulnerability of cancer cells towards viruses and make the viruses into even more effective cancer therapies using those tools of genetic and molecular engineering. So we know, and Katie mentioned this as well, that a lot of cancer cells display certain receptors on their cell surface in large numbers. And we can engineer proteins that bind specifically to these receptors. We can then go ahead and swap out the proteins on the surface of the virus with these proteins that we've made and which bind to the cancer receptors. So we now have a virus that homes in on and selectively infects cancer cells that display these receptors with the match. But most cancer cells also have a bunch of mutations which lead them to producing lots of signaling proteins which boost cell replication. It's all about making more of the cancer cell. And we can take advantage of the fact that the cancer cell is already making these signaling proteins in large quantities by taking from our own genome a bit of DNA from the gene for one of these signaling proteins. And this bit of DNA is known as the gene's promoter, which you may have heard about before. You can think of it as the gene's sort of on-off switch. If we take this switch and we integrate that into the virus's genetic material, then the cancer cell's gene replication and expression machinery recognizes it, and it starts producing the viral proteins in large quantities in the same way that it's already making in the same way that it's making the signaling proteins, which have the same switch. So we now have a virus that selectively recognizes cancer cells, and when it gets inside cancer cells, is much more effective at replicating itself compared to normal cells. So far, so good. But oncolytic viruses have a further advantage over a lot of other cancer therapies. I said that cancer cells that get infected with them make more viral particles, which makes more therapeutic agents in the entire body, and the cell eventually dies. But there's actually quite a few different ways in which a cell can die. So a cell can die through something known as programmed cell death, which is a natural process whereby the cell shuts down, it shrinks, and it dies, and it has very minimal impact on its surrounding environment. But cancer cells which have been infected with an oncolytic virus, they die in a sort of much more violent way. They basically burst open and release all of the cell's contents to the surrounding environment, including those tumor-specific antigens that Katie talked about earlier. And this type of cell death is known as immunogenic cell death, meaning it generates or it stimulates an immune response. The immune system basically recognizes that something has gone really wrong here. And if it wants to prevent that sort of same violent cell death from happening throughout the entire body, then it needs to stop whatever caused this in the first place. So we have a bunch of cells from our immune system flooding to the site of this dead cancer cell, and among other things, recognizing those tumor associated antigens. The process that Katie described earlier of T cells sort of being presented those antigens by antigen presenting cells takes place. T cells mature, they are then specific for those tumor antigens. And we have a host of T cells that can recognize the cancer cells using the antigens. The T cells can fight the live cancer cells that are just in the same tumor site, but crucially, they can also go off and circulate throughout our entire bodies. And if the cancer has metastasized or it's spread through other parts of the body, then these T cells can go off and fight these metastasized tumor sites, which can be really far off. And the result is that we get a sort of systemic immune protection. And once the immune system is primed in this way, it can recognize when the same tumor antigen appears in the future, which provides a sort of constant surveillance and protection against the same tumor or the same cancer reemerging in the future. And it's really the fact that oncolytic viruses have this direct tumor killing effect and this sort of systemic immune boosting effect that makes them really interesting and promising as a cancer therapy. Now, researchers have tried to make this immune stimulating effect even more powerful. I mentioned some of the ways in which researchers have sort of tweaked or engineered viruses to make them into oncolytic viruses. Some oncolytic viruses have also been edited to include genes for proteins that are part of our immune system and which boost the immune response. So viruses that have been edited in this way, when they infect a cancer cell, in addition to the cancer cell making more of the virus, it also starts churning out these immunostimulatory proteins. And once that cancer cell dies and releases its contents, those immune boosting proteins help to recruit more immune cells to the cancer site, ensures that we have a much stronger immune effect against the cancer cell and those tumor-associated antigens. So the systemic boost from the immune system is much greater with a bit of genetic engineering. But when we're using oncolytic viruses to fight cancer, one of the big challenges is actually our own immune system. So although we've designed these viruses to selectively infect and kill cancer cells, our immune system doesn't distinguish between a therapeutic virus and a naturally occurring virus. The moment that we put an oncolytic virus into the body, the immune system will mount a response against it. And that's often before the virus actually gets to the cancer site and gets to have its therapeutic effect. Additionally, if a patient's been exposed to a similar type of virus before, for example, herpes viruses are the basis of some oncolytic viruses, and a lot of people have been exposed to that type of virus, the body might have a pre-existing immunity. So this leads to an even faster immune clearance of the virus because we've already got the memory T cells in our body, which can sort of go off and attack the virus immediately, which makes them, that type of virus, even less effective in that particular patient. So while some immune activity is beneficial, like I said, oncolytic viruses can help sort of train the immune system against a cancer. We need a bit of a delicate balance because too much immune activity can mean that the virus can't do its job, but too little immune activity means we don't get a systemic immune response. And there's some research looking at ways in which we can sort of transiently modulate the immune response or use sort of special ways to get the virus to its site without the immune system attacking it first. And another of the key challenges in using oncolytic viruses is effectively delivering the virus to the cancer site and ensuring that it's sort of distributed throughout the tumor. So unlike small molecule therapeutics, which are very small and tiny and can diffuse really easily throughout the body, viruses are much bigger. And that presents a set of unique challenges. So when you administer an oncolytic virus intravenously, it needs to travel throughout the bloodstream to reach the tumor. But along the way, the immune system can kick in, it can clear the virus, or even organs like the liver or the spleen can just like filter out the virus before it reaches its destination. And because viral particles are large, they are also much less efficient at going through blood vessels than things like small molecule therapeutics. Again, fewer viral particles reach the cancer site. But even when the virus is actually at the tumor site, it's quite difficult to actually penetrate the tumor structure because a lot of solid tumors have this sort of really dense and fibrous structure and also really sort of irregular blood vessel formations, which can make it difficult for the quite big viral particles compared to small molecule therapeutics to penetrate the deeper core. So what you might have is that the outer edges of the tumor might be infected and treated, but the rest of the tumor core really isn't infected at all. So one of the ways around this is to actually inject the virus directly into a tumor site. But as you can imagine, that's not really feasible for all types of cancers, particularly ones that are really deep down inside the body or which have metastasized in different parts already. And using live viruses in cancer therapy also raises some safety challenges, as you can imagine. One of them is just off-target infection. So although we've designed these viruses to selectively kill cancer cells, they could also infect normal cells and potentially cause tissue damage and really bad side effects. And when we think about administering this therapy to immunocompromised patients, then we have a whole other host of challenges because in these individuals, their immune system's much less efficient at controlling viral infections. So instead of being safely contained in the sort of tumor, the virus can sort of start replicating throughout the entire body. You can have a systemic infection, which can be quite dangerous. Another safety challenge is just managing the immune response. Keep kind of coming back to how important the immune system is in therapies like this. So we want the immune system to recognize and kill the cancer, but if we have a really strong reaction to the virus itself, that can cause inflammation or infection-like symptoms. Like it could be simple things like fever or fatigue, but in some cases it could be quite a strong systemic response and lead to things like cytokine release syndrome or other conditions which can actually be fatal for the patient. So if we look at what's actually on the market, T-VEC is the only oncolytic virus to date which has been approved by both the FDA and the EMA, and that was back in 2015. It's a modified herpes virus that's used in patients with advanced melanoma. So that's melanoma that can't be removed entirely through surgery. And it's one of the ones that's actually directly injected into melanoma sites. Interestingly, although we talked about a few different ways in which we can edit a naturally occurring virus to become an oncolytic virus, T-VEC is a good example that you don't actually need multiple of these genetic edits to be an effective therapy. So T-VEC hasn't been engineered to be specific against melanoma cells. T-VEC infects normal cells and cancerous cells just as well. The only difference is that through a deletion in the herpes virus genome, T-VEC's unable to replicate in normal cells, whereas it is able to replicate in cancer cells, which means it ends up killing the cancer cells. And it also encodes for a really powerful immune-stimulating protein, which, as I mentioned, it attracts immune cells to the site of the cancer, encourages a really strong immune response against the dead cancer cells. Although there's only one sort of FDA, EMA approved oncolytic virus on the market, there's quite a few different candidates in different stages of preclinical and clinical development. So each of them uses a different set of edits to make the oncolytic virus. So there's a lot of activity in this area, even though there's only sort of one product on the market. And as we look to the future and the ways in which we can enhance the effect of these therapies, Katie mentioned this before, but one promising strategy is to look at delivery and specifically encapsulation. So this involves packaging the virus into things such as nanoparticles or liposomes. There's a lot of interest on packaging up a whole range of different cancer therapies in this way. And really a lot of attention since the mRNA vaccines developed by Moderna and Pfizer and BioNTech were packaged in lipid nanoparticles in a really effective way, which a lot of people took notice of. And encapsulation serves a few different purposes. So it protects the virus from being attacked by the immune system. It allows more viral particles to sort of survive and get to the cancer. And it also allows the viral particles to go deeper inside that dense, fibrous, solid tumor core, which again enhances the spread, enhances the ability to infect cancer cells. And whenever we have sort of a controlled delivery method like this, it just means that we can dose the treatment more effectively as well, which means we can reduce things like unwanted side effects. So a lot of research happening in this area. And finally, similarly to cancer vaccines, research is also looking at combination approaches for oncolytic viruses. Another thing being looked at is combining oncolytic viruses with immune checkpoint inhibitors, those drugs that take the brakes off the immune system. And it's really a perfect match because the oncolytic virus stimulates an immune response. You want that to be balanced but quite strong. So combining existing drugs in this way can be really robust. But we don't have to combine oncolytic viruses with just sort of advanced immune targeting therapies. There's also benefits to just using them alongside really well-established treatments like chemo and radiation. So these treatments can help weaken the cancer cells and make them a lot more susceptible to being infected by a virus. And additionally, the dead tumor cells as a result of chemo and radiation release the tumor antigens into the tumor site. The oncolytic virus stimulates an immune response against those antigens. So you have a real synergistic effect, which is sort of more than just the sum of the two parts. More broadly, people are seeing a lot of potential in this area. And so we're hopefully going to see more oncolytic viruses reach the market over the coming years. And with that, I'm going to pass over to Louisa to talk about some of the legal issues you might come across in this area.
So thanks to Erik and Katie for taking us through the science. I'm now going to take us all back into our comfort zones a little bit more on the legal issues. I've got 10 minutes, and I'm sure there's a lot of things we could talk about here. I'm a commercial IP lawyer, so I'm going to focus on that side of things. So these are really complex products, right? Cancer vaccines and oncolytic viruses. A single product will incorporate lots of different technologies from lots of different places, and that this sort of on the screen is a selection of the types of technologies you might be talking about. So, for example, with a cancer vaccine, you might have the mRNA and an adjuvant, the delivery system. You might be using gene editing technology, and as a company developing one of these products, you may well be having to license in IP from more than one different third party, and you're having to pay potentially royalties to multiple different third parties on the one product. So a build-up of royalties could become a significant burden for a party developing one of these products. And in each of these deals where you're licensing IP from different third parties, of course, when you're negotiating the economics of those deals, you have this in mind, of course, this context that you're having to be potentially paying royalties to a number of different third parties. But we can also think about contractual provisions in contracts and how those can be used for to help a licensee, to help a company developing one of these products. And I wouldn't be a licensing lawyer without mentioning royalty stacking. So here we go. So I couldn't find a good picture for royalty stacking. So here we have a cute little boy stacking blocks. As a licensee, you might be stacking up royalties payable to third parties. And many of you will be aware of what a royalty stacking clause is in a license. But for those of you who aren't, they are clauses that have been used for many, many years in licensing agreements. They're always there, and the purpose of them is to help a licensee from the burden of having to pay multiple royalties to multiple third parties. And they work by saying, so if I've got a license, if you're the licensor and I'm the licensee and I'm having to pay royalties to you on the IP that you're licensing to me, but then I also, for my product, I have to pay Erik some royalties as well. The royalty stacking clause will operate to say, okay, if I have to pay Erik some royalties on my product, then I can reduce the royalties I'm paying to you by a certain agreed amount. And that's generally the way they work. And of course, they're negotiated quite a lot. No two royalty stacking clauses are exactly the same. And some of the things you can think about as a developer of one of these complex products is how you can get your royalty stacking clause to help you a bit more. So sometimes traditionally a licensor will say, okay, this royalty stacking clause can apply, but it only applies if for me, the licensee, if I'm paying a third party royalties on patents, for example. As a licensee of one of these complex technologies, you can say, well, actually, know-how is really important in some of these products. Materials you might be needing to license in a cell line. As a licensee, I want my royalty stacking clause to apply to if I have to pay royalties on know-how and materials as well, for example. Another way as a licensee of one of these complex products to help you in a royalty stacking clause is just the general scope of the royalty stacking clause. So traditionally, if you're a licensor, you'll say, okay, this royalty stacking clause can apply, but only if the third party IP is necessary in order for me as the licensee to practice the licensed IP. But with what we're seeing more of is the broadening of these kind of royalty stacking clauses. So where they will apply not just to third-party technology that's necessary to practice the licensed IP, but also for add-on technology. So technology that's necessary maybe for the product as a whole, maybe to some of these different aspects of the product, although it might not be necessary for me to practice the particular IP that I'm licensing from the licensor. And of course, before I go on, I just want to say these issues that I'm talking about, they're not just specific to cancer vaccines and oncolytic viruses. They're specific to complex products in general. So, the things I'm saying apply equally to things like cell and gene therapies, other advanced medicinal products, things like that. So I'm going to touch on combination products because both Erik and Katie mentioned that in this area, combination products are important. You might have a cancer vaccine combined with a checkpoint inhibitor, for example. And in the previous talk, Jamie talked about some issues that come up when you're licensing or you're making a product that's combining IP from various different sources. So I'm not going to repeat what he said, but I just wanted to touch on clinical trials and we're seeing more and more clinical trial agreements for combination products. And these can be quite complex and bespoke agreements. And there's a lot of things to think about when you're doing these kind of agreements for clinical trials for combination therapy, where you've got two parties, they're coming together, each owning a particular product that they're going to combine in a clinical trial. And you've got to think about, for example, what the roles of the two different parties are in the clinical trial. Obviously, who's going to be the sponsor, but what's the decision-making authority of the different parties, and what are the parameters around the decision-making? You've got to think about the clinical data that arises, what's each party's right to use that clinical data and to disclose it. Think about the IP. Who owns any IP that comes up in that clinical trial? You might say, there are various different ways of doing it. You might say any IP that relates to your technology solely, you'll own, and maybe the rest of the IP could be jointly owned. That's just an example. If you've got jointly owned IP, you then have to think about, well, what are each party's rights to exploit that joint IP going forward? So there's a lot of other things to think about, which I'm not going to go through because I probably only have about 3 minutes left. But if anyone is interested, we can talk about it afterwards. And finally, I did say I was a commercial lawyer, but I am going to say the word litigation. So, because you can't really talk about mRNA without mentioning the litigation that's going on. I've got on the screen there some of the parties that are involved in the so-called mRNA patent wars at the moment. We've had the CRISPR patent wars, now we've got the mRNA patent wars. Probably the most high profile is the BioNTech, Pfizer and Moderna case. In the UK, for example, there were the High Court recently held that one of Moderna's mRNA patents was invalid and the other one was valid and infringed by the Pfizer-BioNTech COVID vaccine. And last week, but BioNTech and Pfizer got permission to appeal to the High Court on that, and to the Court of Appeal. So we'll see what happens on that. But in that whole context, of course, if you're a company doing a deal in this space, you're doing it in this whole context of this litigation. So there's freedom to operate issues that you're thinking about. And of course, any deal that you do in that kind of context, the deal terms as a whole will be informed by that context. But I wanted to just mention about an indemnity clause. So if you're a licensee, say, doing a deal in this area, as standard practice as a licensee of IP, you'll be asked to give an indemnity to the licensor. So if your ultimate product infringes a third party's IP and the licensor gets caught up in that litigation, you as a licensee will be, as standard practice, asked to be given an indemnity to the licensor, saying, if you get caught up in this litigation, I will cover your costs, and if any damages are awarded against you, I will cover that cost. So, but if in this kind of litigation context, if there is some kind of issue, if you're worried about a freedom to operate issue over the licensor's technology from the start, and this completely depends on the context, but you may be able to have a better negotiating position of some kind of more shared risk there and the indemnity, and to try and reduce your risk under the indemnity if you know from the start there's this freedom to operate issue over the licensor's technology. Of course, it completely depends on the circumstances, but it's something to consider right from the start of your deal. And finally, on the indemnity point, if you are, of course, as a licensee, you always think about the indemnity that you're giving, and there's usually a standard procedural aspects of the indemnity, that section of the indemnity of the contract that is pretty standard. If you are operating in this context, though, where you think maybe there's an increased risk than normal that your indemnity might come into effect, make sure as a licensee to take particular care to make sure that those procedural aspects of the indemnity are favourable to you. So, for example, make sure you have complete control of the claim against the licensor, that there's all the appropriate protections in there for the licensor to cooperate and provide you with the right information, and that the licensor has a duty to mitigate its losses. So that's all I'm going to talk about on the legal issues right now. I think my time is up, and everybody probably wants to go and have some wine. So thank you very much, everybody.